Hello everyone, and welcome to our second lecture on immunity. In our first lecture on immunity, we talked about the innate response, and in this lecture we will be talking about the adaptive response. As usual, you have your lesson objectives. There are quite a few of them for this lecture because there is a lot to understand about adaptive immunity and a lot to understand about how it is different from the innate response. You've seen this slide before in our previous lecture, but I wanted to start with it again, just so, again, we are on the same page when we're talking about the different types of immune responses. We have a basic understanding of what innate immunity is and what adaptive immunity is. So with innate immunity, it is nonspecific, meaning it responds to every microbe roughly the same way each time we see it. And its primary goal is to prevent entry of microbes. And if it can't do that, then what it tries to do is trap the microbe in a specific location so that it doesn't spread too far throughout the body. Now, the adaptive or the acquired immune response, it's also sometimes called the specific immune response, there are a lot of different terms for this, is something that is developed throughout our lifetime. So the innate immune response is something we're born with and functions basically the same way throughout our lifespans. But this second type of immunity, this very broad category of immunity that we call the acquired immune response, is one that develops over time. We are born with the capacity to develop an acquired immune response, but when we are born, we don't have this type of immune response. It's something that we have to develop over time. This type of immune response is required by specific microbes, and its primary function is to cover, and I put that in quotes because it's not an exact function, but it's kind of a good analogy, but it's to cover a microbe with specific proteins we call antibodies and help immobilize or inactivate that microbe in the body. So like I've mentioned previously, the adaptive immune response is one that develops throughout our lifetime. We are born with the capacity to develop this response, but we don't have it at birth. This type of response is unlike the innate response in that it is geared towards specific microbes. Remember, with the innate response, it pretty much functions the same way every time we see any type of microbe. So things like inflammation, phagocytosis, those are going to happen no matter what type of microbe we see, whether it's a bacteria, a virus, a fungus, a helminth, etc. The complement cascade might be better for things like bacteria, helminths, fungi, but at the same time, it doesn't really matter which of those happen to invade us, whether it is species A, species B, species C, or type A, type B, or type C, the complement cascade works basically the same way. And interferons work primarily against viruses, but again, it doesn't matter what virus. Any virus that comes in, interferons are going to be activated. But in the adaptive immune response, the activities, these targeted activities to deal with microbes, focus on just specific types of microbes. As a result, each of us have our own unique kind of immunity fingerprint when we're talking about the adaptive response. Everybody has their own unique adaptive response because every single person on this planet has been exposed to slightly different quantities and slightly different types of microbes. Basically, the adaptive immune response develops in response to exposure to microbes. And because each of us lives a unique life, even, people who live in the same household, even if they've lived in the same household their entire life, if they haven't spent every single moment of their life with each other, then they've been exposed to slightly different microbes at slightly different times. And that differential exposure of certain types of microbes creates a unique fingerprint when we're talking about the adaptive response. Now, overall, the adaptive response requires a few main things to become activated and to function. It relies on specialized white blood cells that have the ability to recognize and respond to specific microbes. Not just microbes in general, but specific microbes. So the innate immune response, again, just treats all of these basically the same. But our adaptive immune response is going to be able to look at each of these and say, oh, look, this one is different than that one, which is different than this one, and different than that one, and different than that one. Because of the ability to recognize the difference between microbes and remember that they have been seen before, our body is able to create a targeted response to that microbe so that the next time we see it, we are able to recognize that it is present, remember that we've seen it before, and act accordingly. Ideally, this ability to recognize specific microbes and remember specific microbes helps with kind of three main things when we're talking about fighting off infections. Hopefully, it helps increase the effectiveness for fighting the infection because we know exactly what we need to do to fight off this specific microbe. Rather than just generally activate some kind of defenses, we can activate some specific defenses. Most of the time, this also helps reduce the severity of the disease because we recognize that we have seen a specific microbe before, and we are ready with specific defenses against that microbe. Most of the time, that means that we can reduce the severity of the infection. Typically, this also helps speed up healing as well because the defense that we have against these microbes, each of these individual types of microbes, is more specific to that microbe. It means that we can target just that microbe rather than mounting a huge immune response that will cause some problems in our cells as well. So one of the things I didn't mention in the innate lecture because it was just getting a little crowded in there, but was the idea that oftentimes when we mount an immune response against a microbe, there's a lot of collateral damage to our cells and our tissues as well. In fact, sometimes our body, in the attempt to deal with some kind of microbe and get rid of that microbe, causes more damage to our cells than the microbe causes to our cells. So with the innate response, things like inflammation, phagocytosis, complement fevers, interferons, etc., those can all do some damage to our own tissues. With the adaptive response, because we're creating a very targeted response, most of the time that response does less damage to our own bodily tissues than the activities of the innate response. And so we're able to heal faster as well because we haven't caused as much damage to our own bodies with this type of response. There are three main things that are needed to create this adaptive response. First are antigens from microbes and what we call antigen-presenting cells, or APCs. There are many different types of antigen-presenting cells from our bodies, and lots of times the phagocytes that we were talking about in innate immunity are the ones that serve as the antigen-presenting cells. The antigens come from microbes. So every microbe has some type of protein or carbohydrate or lipid or some other type of macromolecule on its surface that we recognize as foreign and respond to. Those are the antigens. We also need antibodies, which are proteins that are created by our own body, and then specific white blood cells, especially B cells and T cells. And over the next several slides, we are going to talk about these three main things, what they all are, how they function, and then how all of them come together to create what we call the adaptive immune response. The first thing that we need to start developing the adaptive immune response are antigens. Antigens are substances that provoke the immune response. Typically, they are fairly large. Typically, they're made of protein, but they can also be things like carbohydrates, lipids, etc. And basically, these are things that our body recognizes as foreign. And because our body recognizes them as foreign, we consider them a threat and respond to them in various ways. So when you're looking at this diagram here, you can see all of these microbes here will have different types of antigens because each of them is a different type of microbe, and they will have different types of proteins, carbohydrates, and lipids on the surfaces of their bodies. But another thing that's important to understand is that any individual microbe likely has more than one type of antigen. So if we just kind of zoom in on this bacteria here, we can see that this bacteria has at least 3 different types of antigens on the surface of its cell that our body could potentially respond to. Each one is unique. It's a little bit different than the others that you can find on the surface of that cell. And that is important because the shape of the antigen or the nature of the antigen determines whether or not we can recognize that, respond to it, and remember that so that we can fight against that same antigen, that exact same antigen, again in the future. Now, the next thing we need for our adaptive immune response are antibodies. Antibodies are proteins that are made by our body. They are sometimes also called immunoglobulins, and sometimes they are abbreviated as Ig. So these are, like I said, these are proteins made by our body. They are shaped like a Y, and they have a couple of different important regions on them. First of all, they have what we call heavy chains and light chains. Heavy chains are the longer ones. They make kind of the inside of the Y shape. And the light chains are the external ones, these shorter ones that are found on the outside. Aside from the heavy chains and light chains, we have what's called the constant region and the variable region. So the constant region is basically the same for every single antibody that you will make. It is a protein or a piece of a protein that has the same sequence of amino acids no matter what specific antibody you are talking about. So that's a constant region. It doesn't change. It stays the same throughout your body. The variable region, though, these are the ends that are found on the end of the Y shape. These are the places that are actually going to bind the antigen. The variable region is also sometimes called the antigen binding site. And you can see that at the edge of this variable region, or at the edge of the end of this Y, they have kind of a unique shape. The reason for that is every person, again, in the absence of some kind of genetic anomaly, is able to create potentially millions of different types of antibodies by switching up the shape of the variable region or the shape of this antigen binding site, so that you have one type of antibody that can bind to antigen A and another type of antibody that can bind to antigen B, and then antigen C, antigen D, antigen E, etc. This is how this ability for our bodies to create these different shaped regions at the end of the antibodies, this is how we are able to protect ourselves from so many different types of microbes. Whether that happens to be something like Staphylococcus aureus, or Streptococcus pyogenes, or Corynebacterium diphtheriae, or coronavirus, or influenza, or various helminths that we might run into in our life, or Candida albicans, which is a type of yeast. This variable region is the thing that gives us the capability to respond and remember so many different types of microbes, because they will match up with certain antigens that are found on the different types of microbes that we encounter. Now, the function of antibodies is to attach to pathogens or toxins sometimes and help inactivate them. There are 5 main ways this is achieved. The first is through neutralization. Neutralization occurs when antibodies bind to antigens on a pathogen and prevent that pathogen or that microbe from being able to attach to our cells. This can work hypothetically with any type of microbe, but it works very well with viruses. So, like I said, it works really well with viruses, although it's possible for other microbes too. And the reason it works so well with viruses is because, remember, viruses have to bind to receptors on host cells. If our body creates antibodies, these proteins that are able to attach to the virus, either on the spike proteins or just the capsid itself, then what this does is block the ability of the virus to bind to the host cell receptor. If the virus cannot bind to the host cell receptor, the virus cannot get inside the cell and cannot make us sick. The next primary antibody function is precipitation. Precipitation occurs primarily with toxins, which are oftentimes protein molecules that microbes make that cause some kind of damage in our cells. So there's lots of different potential toxins that microbes can make. And sometimes the toxin is what causes the most damage in an infection rather than the bacteria or the microbe itself. And with precipitation, what ends up happening here is antibodies will bind to the toxin, which again, like I said, is usually a piece of protein, and cause that toxin to precipitate out of solution. And what that means is when a toxin is in a solution, it's basically suspended in some kind of liquid. It's floating through the body, either in our blood or our plasma or interstitial fluid. There are various different types of body fluids. And what precipitation does is these relatively large antibodies will stick to that toxin or to that protein and make it so that it is not soluble anymore. It makes it so that its chemical nature changes and it may get too heavy or it may just have the wrong charges now. But whatever it is, that toxin cannot stay in solution. It becomes a solid. And when it becomes a solid, it becomes inactivated, and our body is able to then engulf that and destroy it, and it won't cause us any damage. Opsonization is our next function of antibodies. And with opsonization, essentially what is going on is that antibodies are binding to the surface of a microbe, and they're making it so that phagocytes are able to come engulf this microbe and grab onto it a little bit better. There are a couple ways this works. Sometimes the microbe is kind of slippery. It may have something like a capsule or a slime layer that prevents a phagocyte from attaching very well to that microbe. And the antibodies basically give the phagocyte another place to kind of hang on to so that it can engulf the microbe. Another kind of way opsonization works is that sometimes, especially with very small microbes, they can kind of get missed by phagocytes. The phagocytes are looking for them and just can't find them because they're extra tiny or they are hidden in some manner or another. And what happens in this case is the antibodies stick to the outside of the microbe and they act kind of like a flag or like a signaling device. And they say to the phagocytes, hey, this is where the microbe is. Come over to this location and deal with the microbe here. The next thing that antibodies do is help with what we call agglutination. Agglutination means sticking things together. And what happens with agglutination is basically the antibodies help the microbes stick together and make big clumps in one location. Since every antibody has 2 antigen binding sites, basically every antibody has a capacity to bind to at least 2 microbes. So in this case, we have a situation where this antibody can bind to microbe A and microbe B, and this antibody can bind to microbe C and microbe D. And what this does is trap these microbes in one location and kind of clump them up together in one space so that phagocytes can come over and engulf a bunch of microbes at once and deal with them rather than having to kind of chase them down. And then finally, the last thing that antibodies do to help get rid of microbes is oftentimes they will help with complement activation or help with a complement cascade. So technically the complement cascade is part of the innate immune response, and that is still true. I'm not changing that on this slide at all or anything. But occasionally, if there are antibodies that exist already, they will kind of take part in this membrane attack, basically because they're around, they're present, and they make the complement cascade more effective, or the complement activation more effective. They make the membrane attack complex work even better to create holes in those microbial membranes. So they oftentimes will help out with that. Now we have just one last slide about antibodies before we can move on to B and T cells and then start putting all of this information together to see how we develop our adaptive response. So far you have learned about antibodies in terms of their structure. They are these Y-shaped proteins with a couple of different important regions on them. The main region that's important, at least to us, are those antigen binding sites, those variable regions at the ends of the arms of the Y. You've also learned how antibodies function. Basically, they work to attach to antigens that are found on the surface of microbes, and they inactivate that microbe as a result. And the last big thing that you need to understand about antibodies is that there are different classes of antibodies. There are 5 main classes of antibodies, and I've got them listed here in alphabetical order, not necessarily in order of importance or in order of amount, just alphabetical order. And the classes are IgA, IgD, IgE, IgG, and IgM. Each class of antibody has their own specific kind of unique characteristics. For example, IgA is a dimer, which means there are 2 of them joined together. It is found primarily in mucous membranes, and its main purpose is to trap microbes. So we find a lot of IgA in our mouths, in our eyes, in our nose, in the upper part of the respiratory tract, and the primary function of that is to help trap microbes before they get too deep into the body. The next type of antibody is IgD. IgD is a monomer, meaning it's just a single Y-shaped protein by itself. It's typically found in very small amounts in most people, and most of the time it is found attached to B cells, which we'll talk about more later. But most of the time it's just attached to the B cell, and it will let the B cell attach itself to some kind of antigen or something that B cell needs to attach to. IgE is our next one. This is also a monomer, and this one is primarily active during allergic reactions and when someone has a worm infection. So if you have a patient who has high levels of IgE, or if you yourself have high levels of IgE, then chances are good that that person or you either have allergies or are having an allergic reaction to something at that moment in time, or you or they have a worm infection. IgG is also a monomer, and this one provides long-term immunity. This one circulates throughout our bodily fluids, especially the blood and plasma, interstitial fluid in some cases, and what it does is provide us with immunity that can last sometimes months to years, to decades sometimes. So this is one that, let's say you get infected with a certain disease as a child, or you get a vaccine as a child, you may have IgG that is specific to that microbe you were infected with or to that vaccine that you received for years to come. So a good example is a measles infection or getting a measles vaccine. You will have IgG to that measles virus for decades, if not the rest of your life. And that IgG will just be circulating throughout your body and helping protect you the next time you come in contact with that measles virus. And then IgM, this is a pentamer, meaning there's 5 of them all attached together in one kind of unit. Its primary function is agglutination, and it is the first antibody that's produced during the adaptive immune response. So we will talk about how the antibodies are actually produced in the immune response in just a few minutes. But for now, just focus on the fact that IgM is a pentamer. It's very good at agglutination because there's 5 of them attached together, so there's 10 binding sites overall. And it is the first antibody we produce in response to a specific microbe. So far, we have talked about 2 out of the 3 main things that we need for the adaptive immune system to develop, or the adaptive immune response to develop. And those are antigens, which are found on the surface of microbes, and antibodies, which are produced by our body in response to antigens. Now, the last thing we have to talk about are these B and T cells, and then we can put all of this information together and see how it works so that we can create those antibodies and respond to antigens. So B and T cells are certain types of lymphocytes. They are white blood cells that are part of the immune reaction. They're made in the bone marrow, and they are found circulating in the blood and lymph for the most part. Now, we don't talk about lymph too much in this class, but just to give you kind of a very broad overview of what that is a system in the body that works in conjunction with various other systems and helps deal with waste and waste products, especially when we're talking about the waste products associated with the immune reaction. So there are 2 main types of blood cells that we need when we're talking about the adaptive immune response, and those are T cells and B cells. T cells are lymphocytes, or these certain types of white blood cells that mature in the thymus, and they have 2 main roles. They kill tumor cells and help activate B cells. We are going to focus on their role in activating B cells. And then the B cells, our other type of lymphocyte that we are going to focus on, are the types of cells that will produce the proteins that we call antibodies. So when we're talking about these lymphocytes in general, we have just general blood cells that are made in the bone marrow. Those can differentiate into various types of blood cells, and we are going to be focusing on the ones right over here, the T cells and the B cells. We'll talk a little bit about the plasma cell as well. Now that you know enough about antigens, antibodies, and B and T cells, it's time to look at how all of these fit together to produce what we call the adaptive immune response. So the adaptive immune response starts to develop the first time we encounter a specific microbe. So we are going to give our microbe a name, and it's not going to be a super fancy name, but it's going to be a name and it will help us keep things straight. So our microbe for this scenario is called microbe A. And the first time we see microbe A, whether we are a month old, 5 years old, 10 years old, 23 years old, 89 years old, whenever it is in our life that we see microbe A for the first time and it gets inside our body for the first time, our innate response is going to kick in. Things like inflammation, fever, phagocytosis, the complement cascade, interferon, those things are going to go ahead and be active in their attempts to get rid of the microbe. Chances are good we are going to get noticeably sick from this infection. because as good as our innate immune response is, it's not perfect and it can't deal with everything. So it's going to do everything it can to get rid of that microbe, but we're probably going to be sick. However, while we are fighting that infection off, certain cells, especially the ones involved in phagocytosis, are going to grab onto that microbe and basically tell our adaptive immune response, hey, we've got a new microbe here. I need you guys to do your job so that the next time we get exposed to this same microbe, we have some extra help with the infection. So the phagocytes involved in the innate immune response are oftentimes what we call antigen presenting cells. Antigen presenting cells are immune cells within our body that have the capability of grabbing onto a microbe, destroying it, and picking apart the individual parts of the microbe, and then taking the antigens from that microbe and putting them on their surface. So the first time we see microbe A and phagocytosis is going on, certain cells that are involved in phagocytosis are going to engulf those microbes, destroy them, and sort of select out the antigens. Once they have selected enough antigens, they take those antigens and they put them onto their surface. And they don't just randomly put them onto their surface. They put them onto specific proteins called major histocompatibility complexes, or MHCs. You read about those a little bit in your book, or at least I hope you did. You don't need to know a ton about them for this class, but basically these MHCs tell our body, this is self, this is you, you don't need to attack it. And they also will signal to the T cells that we'll talk about in a minute, hey, this is your own body presenting you a foreign threat, not just someone random telling you there's a foreign threat. So these cells that we call antigen presenting cells process the antigens from the pathogen, that microbe A that's making us sick, and they take just the antigens, not the whole microbe, just the antigens, and display them on their surface attached to the MHC markers. When they do that, then the adaptive response can begin to develop. Something called a naive T cell comes over, and a naive T cell is just a T cell that hasn't been exposed to antigens yet. And that naive T cell is going to recognize the MHC and antigen complex. What it's going to see is that, hey, this is my own body telling me that there is an invader and a threat and I should do something about it. And this is the specific threat. So that naive T cell takes that antigen and it says, okay, this is the pathogen or the antigen that I need to work against. That naive T cell goes through a series of differentiation events, which just means it matures and changes slightly. And although there are a couple of other things that are not shown here about T cell maturation that I don't have you focus on, the big thing that we pay attention to is how the T cell matures and interacts with B cells, because the B cells are what are ultimately going to get us antibodies. So this mature T cell is going to take that antigen that it had previously been presented, and it's going to display it on its surface. And it's going to say, hey, B cell, come over here and check out this antigen. And then once you've checked it out, could you make some antibodies for us? The B cell is going to come over, attach to that antigen and MHC complex, and it is basically going to learn the shape of that antigen. So it's going to feel the antigen, get an idea of what the antigen is shaped like, and then once it has learned that antigen, it's going to multiply rapidly. So it's going to make a bunch of copies of itself. And from there, all of those copies are going to be able to differentiate into one of two types of cells. The first type of cell that can be made after that is called the plasma cell. The plasma cell is going to be a specialized cell that releases a bunch of antibodies to that specific antigen. So these antibodies are going to be geared towards specifically attaching to the antigens on microbe A. Remember when we talked about antibodies and we talked about that variable region, the antigen binding site? Those antibodies that were produced by this plasma cell are going to fit that antigen that's found on that microbe exactly, because that's what they were designed to do, basically. Now, the other type of B cell that can be produced as a result of this activity is called a memory B cell. The memory B cell isn't going to produce antibodies now, but it can later. So now what we have after all of this, the innate immune system processing that antigen and displaying it to a T cell, that T cell going through differentiation and processing that antigen and presenting it to a B cell, that B cell making a bunch of copies of itself and then going through some differentiation. We have antibodies from the plasma cell that will be able to circulate in things like blood and plasma and sometimes interstitial fluid and potentially other bodily fluids as well, so that the next time we encounter microbe A, we have some immediate protection. And then we also have memory cells that are going to circulate in our body so that if we don't quite have enough ready-made antibodies available the next time we come in contact with microbe A, they can kind of really quick switch on some protein production and create more antibodies to deal with that infection. Now, the next time we encounter microbe A, whether that is a month later, a year later, 2 years later, a decade later, we have these plasma cells and these memory B cells, and hopefully some free antibodies circulating around, that can go ahead and aid in the defense of the body. So the next time we encounter microbe A, the antibodies from the B cells that were created after the first exposure are going to be used to attack the microbe. They are going to do things like opsonization, neutralization potentially, precipitation, all of the different things that the antibodies do to help attack that microbe. What that does is help out the innate response. And so it shortens the amount of time our body needs to respond to the threat. It allows for a targeted response. And oftentimes it will prevent symptomatic disease. There are lots of times in your life that you have been exposed to microbes a second or third time in your life, and you didn't get sick from them because of the antibodies that you had from a prior infection. So the way I like to explain this one is, you know how little kids are always getting sick? They're always getting sick because they haven't had a chance yet to develop their antibodies. And then as you get older, certain things do change with your life. You tend to not put everything that you find in your mouth, and you tend to wash your hands more frequently, which is great. That does help you not get sick as often. But also, one of the reasons you don't get sick as often as you start to get older is because you've had all of these previous exposures to microbes, and you've developed a bunch of antibodies and a bunch of memory cells. And that way, when your little cousin comes over and is crawling all over the floor and spreading a bunch of microbes around, even if you get exposed to those microbes, chances are good you're not going to get sick because you've probably seen them before. And so you already have the antibodies to aid in that immune response. And it helps make things go a lot faster. You may not even notice you're sick, and you can just kind of go on with your life. On this slide, I just have a zoomed in version of that picture I was talking about on the previous slide, on the last slide. And just so you know, this slide order is a little bit different than the order found in the files that you may have downloaded from the text. I apologize for that. I didn't catch the mistake until I had already had the files uploaded into the textbook. So I just changed the order of the slides slightly. It doesn't change any of the information. You may just need to rearrange the slide a little bit on your end, or at least the order of the slides on your end. So again, just to recap what happens with the development of the adaptive response, it starts when the innate response recognizes that there is an invader in our bodies. All of the normal innate response processes go on, and during phagocytosis of the innate response, certain phagocytes are able to become antigen-presenting cells. One example here is a dendritic cell. That is one type of antigen-presenting cell, although there are a couple others. So these antigen-presenting cells will not only destroy the invading microbe, but they will kind of pick it apart and separate out the antigens from all the rest of the microbe. They will take those antigens and they will put them on their surface. They will add them to what we call the MHC, or major histocompatibility complex markers, that tell our body, this is us, this is self. And the T cell, once it realizes that there is an MHC marker with an antigen on it, is going to come over and attach to that antigen and say, oh, okay, this is something foreign that my body is telling me, with the MHC markers, my body is telling me I need to do something about. So this T cell, it's called a naive T cell, it goes through the process of differentiation, which basically just means it matures and changes a little bit in that process. This T cell is then going to take that antigen and display it on its surface. It's going to display it and say, hey, B cell, I have an antigen. Can you come over and check it out? The B cell is going to come over, attach to the antigen, learn the shape of the antigen, and make the correct antibody as a result. That B cell is then going to make a bunch of copies of itself. Some of those copies are going to go on to become plasma cells, which will release antibodies into bodily fluids. And some of those copies are going to go on to become memory B cells. Those memory cells will not produce antibodies this time, but they will circulate throughout the body. And if in the future we don't have enough antibodies from the plasma cells, those memory cells can go ahead and very quickly produce more antibodies to help fight this infection. So once the plasma cells are producing and releasing antibodies and the memory cells are circulating, the next time we see a microbe, we have antibodies. We see that same microbe, not just any microbe, but that same microbe that had that antigen that we responded to. The next time we see that same microbe, now in addition to the normal innate immune response activities, we have antibodies that can help. This helps shorten the amount of time that we are infected. It will hopefully help prevent symptomatic illness. Hopefully you won't even know that you've been exposed to this microbe. And it just, in general, helps reduce the impact of the microbe on our bodies. So there is just one more thing we have to talk about when it comes to adaptive immunity, and that is the different types of adaptive immunity we can have. Now, all of these, everything that I'm saying on this page, is a type of adaptive immunity because it deals with either the creation of antibodies inside the body or the exposure to antibodies from some other source. So when we're talking about adaptive immunity, we can either have natural immunity or we can have artificial immunity. Natural adaptive immunity is acquired or developed just through normal human activities, whether it is getting a cut or being a developing fetus or a breastfeeding infant, whatever it is, just normal human activities. Artificial acquired or adaptive immunity is something that's acquired or developed through some kind of a medical intervention. So something we do in a hospital or a clinic that gives us this type of immunity. Active adaptive immunity comes from being exposed to antigens, which allows our body to make antibodies and gives us long-term protection. And passive adaptive immunity is developed when we are exposed to antibodies, and basically our body is injected or exposed to pre-made antibodies and gives us short-term protection. So I will warn you that these topics can be challenging for people. This is definitely something to spend some time with because it's about to get a little bit more complicated and we're going to start to put these together too. So we can have natural active immunity. We can have natural passive immunity. We can have artificial active immunity, and we can have artificial passive immunity. So let's just start with natural active. Natural active immunity occurs when we do something that is normal to a human. We are exposed to antigens in the process, and our body responds by making antibodies. So this person has gotten a cut. While they got that cut, they were exposed to microbes. At that time, their innate response is going to kick in. The innate response is going to go ahead and activate that adaptive response. The adaptive response is going to develop, and that person's body is going to produce antibodies to the antigens. So I'm just going to go ahead and write a few things there so that you can write some things in your notes. So the cut that they got introduces some microbes. The microbes have antigens. And this person's body is going to respond by making antibodies. So that's one of the key things about active immunity is that we are exposed to the antigen and we make the antibodies. The next type that we'll focus on is natural passive. So there are really only two main times in our life that we acquire natural passive immunity. And that is when we are developing as a fetus and also during breastfeeding if we are breastfed. So during natural passive immunity, the way this works is that either during fetal development or during breastfeeding, the mother's body makes antibodies and passes those antibodies off to the infant. So this differs from active immunity because in active immunity, the person's body, in this case the baby's body, would be exposed to the antigen and respond by producing the antibody. Instead, in this case, it's the mother who has been exposed to the antigen and has produced the antibody and now is sending just the antibodies, not the antigen, to the baby. Those antibodies will then circulate in the baby's body for a relatively short period of time and give the baby the same type of protection that the mother has. So again, this is natural because this is just part of being a human, but it's passive because the baby is not going to make antibodies themselves. They are not being exposed to an antigen and are therefore not going to produce the antibodies. They are just being given pre-made antibodies to help protect them. Because those antibodies are pre-made and they don't have to make them themselves, it lasts a relatively short amount of time. So a couple months maybe would be the longest that type of immunity would last. Now our next type is artificial active. Artificial means we're using some kind of medical procedure, and active means we're forcing the body to make antibodies itself. So in this type of immunity, we expose someone to an antigen and force their body to make antibodies as a result. The most common type of procedure that would be used to have someone develop artificial active is through something like vaccination. So in vaccination, we give someone an antigen from a microbe. And as a result of that antigen being introduced in the body responds by producing antibodies. So if you're curious about vaccination, we're actually going to have an entire lecture. Our next lecture is all about vaccination. But for right now, it is a great example of how someone would develop artificial active immunity. Someone is exposed to the antigen of the microbe. It's put into their body recognizes that antigen as foreign, goes through that whole B and T cell development process, and produces antibodies as a result. And then our last one is artificial passive. This is again acquired through a medical procedure, and instead of forcing the person to make their own antibodies, we are just giving them pre-made antibodies. Sorry, that is artificial passive, not active. So in this situation, the antibodies are typically made somewhere in a lab. In the past, we oftentimes had to make these in like animals or other humans or something like that. We still can that way as well, but these days we have the capability, oftentimes at least, of making these in a lab relatively quickly, easily, and safely, as opposed from having to get them from an animal or another person. And then those antibodies themselves, once they've been produced in the lab or gotten from a person or another animal or something, are just given to a person either through something like an IV or injections. Typically, those would be the ways that we would introduce them into the body. This type of immunity is oftentimes used to prevent infection when someone's been exposed to some kind of very dangerous agent that they probably don't have antibodies to. A good example is rabies exposure, because the majority of humans in the world do not have antibodies to the rabies virus. And when they get exposed to it, it's very important to protect them from that infection. And there's not necessarily time to vaccinate them after exposure. So what we do to make sure that they are protected and won't get sick from the virus is that we just give them pre-made antibodies that will help take care of the immediate infection. And then later we can decide if they warrant some kind of vaccine to help protect them from any lingering virus that may be in the body. This technique is also one that's being used to treat COVID in many cases. When you hear about people receiving monoclonal antibody treatment, this is what is happening. They are receiving antibodies to COVID, and those antibodies are going to fight the infection. And this is a form of what we call artificial passive immunity. Those antibodies that they're being given are antibodies against COVID. They are going to go into the body, attach to the virus. They're going to neutralize it so that the virus can't get into cells and infect them. And that is going to give the person at least a little bit of protection against that virus immediately. However, because that person did not make those antibodies themselves, that protection isn't necessarily going to last very long. If they want longer term protection, their body's going to have to make its own antibody to the virus. So again, just to summarize this slide, we have 4 different types of adaptive immunity that we can develop throughout our lifetime. We have natural active, which is a type of immunity that we get through just everyday human activities, and we are exposed to an antigen and our body responds by making antibodies. We have artificial active, where we get this through some type of medical procedure. We are exposed to an antigen and our body responds by making antibodies. We have natural passive, where a mother will make antibodies and pass them off to her baby either through fetal development or during breastfeeding. And we have artificial passive, which occurs through some kind of medical procedure where the antibodies are made in a lab or in another person or in an animal. Those antibodies are then harvested, put into some kind of solution, typically, and then given to a person either through an IV or some kind of injection. Those antibodies will work against a disease immediately, but they won't last very long. So in the meantime, if the person wants to protect themselves against the disease, their own body is going to have to kick in and start making some antibodies. Finally, just like with the innate immunity lecture, I have a summary slide here for you all. You can go ahead and read over this. I'm not going to read it to you just because I know you all know how to read, and that will hopefully help kind of crystallize the key points for this lecture and help you understand what the overarching or the big concepts are from all of this information, so you know how to kind of create a scaffold for the knowledge or organize the information in your head.